Okay, so two students have joined me. Okay, no worries. We will start our class. So just want to touch with you basics of lipids. Yesterday we were talking about density. Now a little bit. This we have discussed this in our first lecture, but it's let's discuss it a bit more about lipids. So lipids, as we know, they are very essential. They're the energy source where the lipid could store. They are insulators, a structural role that they have like endocrine function, nervous system, and they also help in the vitamin absorption. So they are divided into four parts. That is simple, complex, derived, and miscellaneous. Simples are fats and oils and waxes. Complex includes phospholipids, glycolipids, sulfolipids, and lipoproteins. And the derived one includes your fatty acids and alcohols, and the miscellaneous aliphatic hydrocarbons and terpenes. Right. So compound one, the compound lipids, or we can say the complex lipids. This one. So the phospholipids, glycolipids, sulfolipids, lipoproteins. So actually they are the esters of fatty acids with different alcohols but carry in addition other substances such as phosphate, nitrogenous base, carbohydrates and proteins as we have discussed before. So normally our this is glycerol towards the three fatty acids group are attached via the ester group. So instead of on the third carbon you have now phosphate group with another group attached to it, making it phosphatic acids and so on. So this is phosphatic acid. On third carbon, your phosphate group is attached, and your alkyl chain side chain is R1 and R2 outside the fatty acid. But if the R1 and R2 is present before, then your fatty acids, your carboxyl group is present, then it's become a phosphoglyceride, right? Some example in the summary that we have discussed on the first day of the lipids: the phosphatidylcholine, phosphatidyl ethanolamine, phosphatidylserine, phosphatidylinistyl, phosphatidylglycerol, diphosphatidylglycerol. So PC, PE, PS, PI, PG, DPG. So all these structures we can see three carbons are there. This carboxyl group with the side chain, phosphoric acid is attached, then choline is attached here. This is choline. This is choline. This is ethanolamine. This is serine. This is inositol. Inositol. This is glycerol. This is diglycerol. So there is one glycerol here. Then again phosphate group is dashed. Then again, so double chained. So as we know, this pure phosphoric acid, these group attached and they become phosphatidylserine, cephalin, lecithin, inositol, glycerol, and so on. So phosphatidylethylamine, so glycerol is there, so towards three carbons, there's ether linkage, there is this one, and there is this ethylamine, different carbon atom. Then phosphatidylcholine, this PC that we have just discussed, this atom is there attached. Then according to your phosphatidylcholine, it could be divided into two parts. First is sphingosphene. So here we can see there's 12 carbon atoms here, and CH3, CH double bond, COH, CnH2, and CH2H. Yeah, this is different substance then. So we can see this. There is one in sphingosine. This is the polar group mostly, which has phosphoridyl base attached to the ceramide at carbon 1, and then it is attached to the ammonia, then carbon double bond with this fatty acid, and this whole is known as ceramide. And cardiolipins, they are diphosphotidylglycerol. So there is glycerol. So towards two uh, carbon atoms, one is phosphate, phosphate, then your phosphatic acid is attached on both sides. These glycolipids, this we have done. Then structural role, what other roles are? Their role in the blood coagulation, as we had discussed. They are required at the stage of conversion of thrombin by active factor X, and activation of factor 8 by activated factor 9, and role in the absorption, lipid absorption in intestine. So lecithin lowers the surface tension of water and aids in emulsification of lipid water mixture. And the role of transport of lipids from intestines. So exogenous triglyceride is carried as a lipoprotein complex, chylomicrons, and which phospholipids take an active part. Also transport of lipids from liver, role in electron transport, lipotrophic action of lecithins, and membrane phospholipids. And insulation, your phospholipids of myelin sheaths provide the insulation around the nerve fibers and provide communication between the hormone receptor on the plasma membrane and intracellular calcium reserve. Yeah, that's it. Some prostaglandins functions, pathological and psychological, physiological. So pathological is like fever, myocardial ischemia, asthma, thrombosis, hypertension, ulcer, diarrhea. And physiological, temperature control, ANS transmission, bronchial tone, blood pressure, and so on. So that's just want to attach something related to with the lipids here. So I don't want to go in details in this part. So this much is enough for us. Okay. Now we switch to what do you say? Nucleotides. So something about nucleotide first. So they are the nitrogenous base, as we know. They are planar, aromatic, and heterocyclous. They are purines and pyrimidines, and number of basing is unprimed here. So like starting in this purine, starting from this nitrogen, 1, 2, 3, 4, 5, 6, then 7, 8, 9. In this case, it's starting from this bottom one, 1, 2, 3, 4, 5, 6. So remember this priming, this numbering in this purines and pyrimidines. So purines is your adenine, guanine, cytosine, pyrimidine, cytosine, thymine, and uracil. Main difference between thymine and uracil is of the alkyl methyl group. On here, 1, 2, 3, 4, 5. On the fifth carbon, a methyl group is attached. That's the main difference between thymine and uracil, right? And in the adenine and guanine, the main difference is that on the third carbon, your main group is attached there, and here it's second carbon it is attached, and additionally your oxygen is added to it. Then there's sugar. So this is 5 carbon sugar. Number of sugars are primed also. 1, 2, 3, 4, 5. And sugars is of two types: deoxyribose and ribose sugar. This is in RNA. On the second carbon, hydroxyl group is absent. On this, it is present, and is absent. Nucleosides are the one without phosphate group. Those result from linking one of the sugars with purines or pyrimidines through the glycosidic linkage and glycosidic linkage. So purines bond with carbon 1 atom of the sugar at the N9 carbon, and pyrimidines bond at the C1 carbon of the sugar at the N1 atom. So on first carbon, this is N9 carbon, and they're having this glycosidic linkage. And phosphate group are also present, mono, di, and tri. So they are bind to the third carbon and fifth carbon of the sugar. So like here, so fifth carbon is adenosine monophosphate, and this is deoxy. This is with the hydroxyl group attached to it, and you have your adenosine attached to it. And in this case, thymidine is attached, and your one phosphate group is attached, and it's also without oxygen. So this is a deoxyribonucleotides, and this is a ribonucleotide. So here we can see the results from linking one or more phosphates with the nucleosides on the 5' end of the molecule through esterification. So nucleotides are the one which are having the same sugar and your phosphate group and your nucleoside base group. All together they make nucleotides. So RNA is a polymer of ribonucleotide, and DNA is a polymer of your deoxyribonucleotide. So both deoxy and ribonucleotides contains adenine, guanine, and cytosine, and ribonucleotides contain uracil, and deoxyribonucleotide contains thymine. So monomers from nucleic acids polymers. So nucleoside triphosphates are important energy carriers, that is ATP and GTP. And important components of coenzymes are FAD, NAD+, and coenzyme A. Some naming conventions. So nucleosides, they are the purine nucleosides which ends with the sine at the end. So like adenosine, guanosine. And the pyrimidines, they end with the dine. So thymidine, cytidine, uridine. Whereas the nucleotides start with the nucleoside name, and we can add mono, di, triphosphate. So adenosine monophosphate, cytidine triphosphate, deoxythymidine diphosphate, and so on. So that was just to give you basics about nucleotides and nucleosides, the difference between them, how they are all different. Okay. Now let's go to the biosynthesis of it. So basically, as we know, as we have just discussed a little, our basic introduction towards nucleotides. So how they are differentiated from nucleosides and how important they are, and also they are very biologically very important in our body. So they are building blocks of our nucleic acids, which involved in energy storage, muscle contraction, active transport, and maintenance of ion gradient. And they are activated intermediates in the biosynthesis, that is UDP glucose and S-adenosine methionine. So they are components of coenzymes, that is NAD+, FAD, FMN coenzyme A. And they are metabolic regulators, that is second messengers, CMP, CGMP, and phosphate donors in signal transduction, this is ATP, and regulation of some enzymes via adenylation and utilization. So these are the one which have intracellular concentration of these nucleotides are highly tightly controlled. So different cell types have different needs and ability to synthesize nucleotides, and concentration of ribonucleotides are much greater than the deoxyribonucleotides, that is 10,000 more than that. So DNTP's concentration markedly increased during the S phase of cell cycle. So imbalance of DNTP concentration may affect the polymerase fidelity also. So indeed they are quite important. So that's the basic structure of your nucleotide. Here we can see is a phosphate group, pentose sugar, and purines and pyrimidine base pairs attached by the glycosidic linkage, and here with the ester linkage, phosphodiester bond. Then there's a nucleobase structures. This is purine, adenine, pyrimidines. Purines includes your adenine, guanine, also hypoxanthine and xanthine. These are new structures. Then thymine and uracil and so on. So there are two major routes for nucleotide biosynthesis. Basically, the first is the salvage pathway. Second is the de novo pathway. If you have activated ribose in the form of PRPP and you have your base attached to it, so it will give a nucleotide and which will further give your DNTPs. And then according to the de novo pathway, we having these activated ribose PRPP plus amino acids plus ATP plus carbon dioxide. So they will give the nucleotide, that is DNTPs. So just remember these two different nucleotide biosynthesis pathway. That is, there is a salvage pathway, then there is a de novo pathway. The salvage pathway includes your activated ribose plus base, giving rise to nucleotide and then DNTPs. On the other hand, the de novo pathway with the help of activated ribose sugar plus amino acids plus ATP plus carbon dioxide, giving you nucleotides and DNTPs. So some synthesis of pyrimidines here we will be discussing. Yeah. So we having this bicarbonate HCO3. We having glycine plus ATP added with glutamate 2-ADP plus phosphate, and then carbomolyl phosphate is produced, leading to the production of aspartate, and then carbomolyl aspartate is produced, giving rise to dehydroorotate. And then with the help of NAD plus you have your orotate, and then from orotate we make orthodiene monophosphate. Orthodiene monophosphate in the presence of PRPP. Then from orthodiene monophosphate OMP, your unimonophosphate is produced, orthodiene, and then CMP is produced further. The same way, this was the synthesis, that how a proper your pyrimidines are produced, right? This is uracil monophosphate and this is cytidine monophosphate. So the same, your CMP and UMP, cytidine and uridine monophosphate, how we can break them? So how the degradation of these pyrimidines are happening in the body. The first phosphate group is removed, leading to formation of cytidine, and then in the presence of water, ammonia is removed, making it to uridine, or directly UMP become a uridine, and then from uridine, your sugar is removed, and then it's become uracil only. Then from this uracil, in the presence of NADPH, it's giving rise NAD plus, and hence you have your dihydrouracil. And then we add this water to it. After the addition of water, it's become uroiodopropionate. Then further it's become beta-alanine, then melanonate semialdehyde, then acetyl coenzyme. So in this way, your whole pyrimidines will be degraded. So what are the defects in the de novo pyrimidine biosynthesis lead to clinical diseases? So how they are related to diseases actually? So first is orotic aciduria. So in this you have anemia, growth retardation, and orotic acid excretion. And causes are it defects in the phosphoribosol transferase or orthidine decarboxylase. And this is orotate here, and then there's a 5-phosphoribosyl 1-pyrophosphate, that is the PRP. So one orotate and these mix together, then PPI is removed, then it's become orodilate. Then you have rudolidilate. So in these conditions, patients are fed with uridine. That is, you give rise to UMP, UDP, and then triphosphate. And this UTP inhibits carbamyl phosphate synthase too, preventing the biosynthesis of accumulation of orotic acid. Just a second. फोन दो दरवाजा बंद कर दो मेरा Now further UTP inhibits carbamyl phosphate synthase too, preventing the biosynthesis and accumulation of orotic acid. So you have a glutamine plus the bicarbonate plus ATP, and then in the presence of PRP you have carbamyl phosphate. Then carbamyl aspartate is produced, then OMP, UMP, UDP, UTP and CTP. So this UTP will be the actually negative feedback inhibitor and will interfere with the enzyme production of PRP. It will not let PRP work properly. So in this way, it will interfere with that. So in summary, we have discussed that pyrimidines are synthesized by de novo and salvage pathway. I mean, de novo means here something related with, in medical term, we use this from the beginning, from the starting. And the pyrimidine ring is synthesized from pre-assembled ingredients, that is carbamyl phosphate and aspartate, and then attached to the ribose. Then pyrimidine biosynthesis is tightly regulated by feedback inhibition. So CTP synthetase, carbamyl phosphate synthetase, aspartate transcarbamylase, and transcriptional regulations all are being taken care in that. And the mammalian enzymes are multifunctional, example carbamyl phosphate synthetase and UMP synthetase, and form multi-enzyme complexes to increase the efficiency. So drug inhibitors of pyrimidine biosynthesis are under development as potential antimicrobial and anti-cancer agents are produced. So that's it from the nucleotide, nucleic acids production and degradation. I should share this also. So students, we are done with the structural of DNA in proper way. We have discussed introduction of biochemistry. We have discussed amino acids and classification of amino acids. We have discussed, we have finished with the lipids in details so far, and we have discussed about nucleotide synthesis and degradation also today. So like pyrimidine biosynthesis basically. So all in all, we are finished with this initial unit of these things. Um, so I can show you some gel electrophorces today. How gel electrophorces is done practically in the lab. That is one of the virtual lab that was told in this content. So we have not discussed any gel electrophorces, I'm pretty sure. So we have discussed PCR very well. We have discussed DNA extraction very well. Now we were discussing from the previous days some basics of your biochemistry, all the uh, basic biomolecules. Okay. So some of the blotting techniques. So remember snowdrop is S for your southern, N for northern, O for nothing, and W for western. So DNA, RNA, and protein. That is drop. So basically, these gel electrophoresis techniques are done in order to identify your various nucleotide nucleic acids, how much they are present in particular sample. So in southern, it's mainly done to target DNA fragments, like for section fragments of gel electrophoresis. It was discovered by E.M. Southern. So in this technique, we exploit the property of radio level probe with a single stranded DNA. And if we want to detect the presence of specific sequence in our mixed DNA sample, then we will accordingly design this probe, which will have complementary sequence to our target sequence. So in this process, first we run our samples over the gel, and then we transfer this gel over the nitrocellulose membrane, so that all the all the proteins or the DNA would be transferred to that membrane via capillary action. And then once it is done, then you add your radioactively labeled nucleic acids corresponding that you're looking for in a sealed container, and then you run this over the auto radiograph in order to see your bands. So these are hybridized nucleic acids, and this is the size markers. So in this way, the southern blotting basically works. So there are various applications of southern blotting, like to analyze genetic patterns in organism DNA, to discover gene mapping, to have a particular DNA sequence present in the genome of DNA, monoclonal leukemia population, sickle cell mutations, and so on. So let's see, there is no voice behind it, but you will see how in this titles that how we do this technique practically in the lab. Just go through the downwards. So in this way, this is done. The same way, in the same technique, your RNA synthesis, your RNA, sorry, northern blotting will be done. So in just instead of DNA, we use RNA as a sample. So RNA is extracted from the cells of interest, but precaution must be taken in order to avoid degradation of our single-stranded RNA by ribonuclease, which is found on the skin and glassware. An addition of also diethyl pyrocarbonate (DEPC) is added, which inhibits the ribonuclease activity, and also baking at high temperature could destroy this ribonuclease activity. So the same way, everything is same. So, but I will show you how it is done. It's a little bit different because it will have some another reagents initially, but it's a formaldehyde agarose gel electrophoresis. We discuss about other the third one. This is Western blot tomorrow in details. So for now, from this link you can download this. So are purines and pyrimidines different from proteins? Actually, they are building block of proteins. So from them, everything is being made. Activated means the things are activated. Your action is activated, which was not before activated. It's become activated later.